Hello, and welcome back to Completely and Utterly Spore-ing, a podcast about bacterial endospore formation, where you may not have come in with any understanding or care for spores, but hopefully by now you care a little bit. I'm still Kit. I'm still here. Callum's still here. Callum's still Callum. Callum knows about
I'm
spores a bit.
I'm still here. I'm not quite spored yet. We've done 3 episodes, and we're only just getting to your research, but that's what it takes. Though we need we needed the context that we've had so far. It's been very interesting. We've learned about I think the reasons behind what your research is actually about, and the, as I say, the context behind it. And so now we're getting into, I guess, the main stuff.
Yeah, I know that it would probably have been easier to start off with actually what I was up to, but the thing is, it makes a lot of the design and stuff, the way that my project was structured, I didn't understand it without all this context. So I don't see why anyone else would understand it
So
without all
we
this
would have
context.
we would have no chance, pretty much. You could have started it with this episode, but no one would have any idea what you were talking about.
Yeah, probably. There are some things that have come up naturally as I've started to explain things because the way I understand things comes from doing my project. So some things will feel familiar because of how they've, I guess, come up in previous episodes, but hopefully it
Yeah,
should be interesting to see how it fits together.
definitely.
So, yeah, from what I've said before, my project was investigating commitment, and it was a slightly non-traditional approach because my PhD student, the person who was supervising me, was working on commitment as her kind of PhD project. That's what her thesis is on, and she'd spend a lot of time picking out maybe genes that she considered promising because of their function and then deleting them and seeing if there was any change. And she'd spent quite a long time on this, found a few things that worked, but was kind of running out of options, and trial and error does wear off, I guess, after some time.
Yeah,
So the way that I was working, the concept of backing them into a corner and seeing how they mutated themselves, forcing them into a situation where the only mutations that could occur were in the genes that we were interested in finding, was kind of the best way to quickly generate information that might help move forward with the area of research, I guess.
yeah. So far we're almost kind of narrowing it down to what specific genes we're looking at. So as we've covered in previous episodes, we're not looking. I think I'm correct in saying we're not looking at. So sigma E has been deleted. Sigma F, we're not allowing it to remove sigma F,
Yes.
because of that particular antibiotic resistance. And then once sigma F is activated, this activates about 100 genes underneath that. We've also deleted 3 genes that we know are involved in commitment, but we're not interested in because they've been very well documented. And so we're looking at some other genes that are part of this 100 or so that are under sigma F, that we're basically, that I say you were looking at how they were involved in commitment.
Yes. And the idea here, the way to kind of promote not only mutants, but to kind of the phrase select for is the phrase that we use. We're kind of selecting for traits that we are interested in, and that is a term that comes from, in terms of biology, the kind of study of evolution, the idea that you have selection pressures and then survival of the fittest or the most suited to an environment. And this obviously is true in the real world with natural pressures, and then our kind of the way that we designed it was loosely based or quite inspired by there's a experiment by someone called Richard Lenski, and he did for decades a continuous culture of just the generations and generations starting from the same colony of a bacteria and seeing where mutations spontaneously happened. So they
Okay.
after a large number of years had a spontaneous mutation where some of the cells started to use a different metabolism, and that was spontaneously created.
Wow.
And then the problem was, because of the fact that it wasn't, the conditions weren't necessarily promoting it, and a random sample was taken every day to form the next culture, it was also spontaneously lost again, which is interesting. But it was the concept of forcing or simulating a evolutionary pressure by backing them into a corner and then maintaining that pressure, because you're not just growing them like that for a day. You're growing them like that for a day. The ones that survive are then being put back in the same conditions, and you're sort of, every generation should select further and further towards the characteristics that you're interested in. In biology we would say phenotypes, which means
I know that
kind
term.
of physical attributes.
Biology, yes.
Yes, yeah.
Yes.
So because you have genotypes and phenotypes, so we don't know what the genotype is, or at the point in my project, at no point do we know which genes are changing, but we know that the characteristics, the behaviors that we're interested in are changing, so we know there must be a genotypic change.
Yeah, and I think last time we really got an appreciation for actually how difficult it is to keep these bacteria in such harsh conditions for so long, because what we talked about last time is when you're selecting for these, or when you're narrowing down to actually just make the mutants that you want, which, as we mentioned, were, things like deleting sigma E, deleting these 3, and keeping sigma F on, because you have to test for each one of those at any one time and then measure to see if it's okay. Has that happened? And then when you make a new generation, has that stayed? It's really difficult. And so I think the main thing I took from last episode is just an appreciation of how difficult it is to even just get to the starting point of your experiment. So it was very impressive.
What was nice was we wanted it to mutate. Once it got to the start of the experiment, we were no longer upset if it started mutating. So we could make the strain that we decided to use, so the triple mutant with the deleted sigma factor, the constitutive is the word we'd use, where it's got forced sigma F and no sigma E, and keeping it, once we'd made that, it could be a stock of it, could be frozen, and then you could basically just take a bit of that at any point and it would start growing, and it would be a clone, because it was from the same colony as the one that we knew was right. It would have the same characteristics that we wanted. So we could use that whenever we wanted it. But the point of the evolutionary kind of experiment, we called it the continuous culture, the idea that things were allowed to mutate. We wanted them to mutate, and we were able to almost control those mutations, not control those mutations because we weren't setting them, but guide those mutations, kind of push them in the right direction by creating these selection pressures that we did by the conditions that we put them in.
At the end of the day, evolution is just an organism responding to its environment and adapting to its environment. And so if you're able to change the environment, then you're able to guide it to the specific changes essentially it needs to survive, in a way, is what you're saying.
Yeah. So we had this kind of, I called it continuous. You'd put this mutant that we'd created would be grown in nutrients until it got to a certain cell density. So in liquid culture, so you grow it in sugary water, but not the sugariest water. It was kind of neutral media. It had all the things it needed, but it wasn't like living the high life, I guess you could say.
Yes.
So it was in this kind of standard media, and it would be grown until there was a certain number of cells. So you can check how cloudy something is to know roughly how many bacterial cells are in there, and you wanted it at a certain density because that means that they're at a certain phase of growth. When bacteria are in a liquid, they kind of lag, and then there's an exponential phase, and then they kind of flatten out, and there's plateau stationary phase, and then they start to die off because they run out of space or nutrients. So in order to make things sporulate, you want to take them when they're in the exponential phase of growth and starve them. So we had to grow things
Right.
So we would take our sample overnight, and then we would transfer it to media for 2 hours, and then we would transfer it to sporulation media, the salt water, for 2.5 hours.
Yeah. So so here you're talking about, before we even get to, because obviously we've been talking about the kind of the sigma F pathway leading to the genes and then sigma E. So this is in terms of the progress of the bacteria and the sporulation process. Here you're talking about at the very beginning, where you've got the normal bacteria, they're at this exponential growth phase, and then you're putting them into salty water, which is essentially removing all of their nutrients, and that's how you started the sporulation process for these bacteria, or these mutants.
Yes, we allowed them to have healthy and normal growth until there were enough of them, and they were in sort of a active division phase, I guess. So they're actively replicating, and then we put them straight into the starvation media, the salty water, and that means that they start triggering the sporulation pathway, and that would be sigma F would be activated, and then they'd be getting stuck at sigma E. But because of the fact that we only did 2.5 hours, there's not enough time for them to start dying because they're stuck at sigma E. They're just turning on sigma F. They're starting to form the spores, but they're not getting, and they're getting stuck because they can't go any further, but they're not getting stuck and then unhappy that they're stuck in the same way, because it's just but so 2.5 hours was spent in the salty water, and then at the 2.5 hour point we would add antibiotic for half an hour,
and this is to check that these different genes that you've deleted in the sigma F thing, it's to test for those, essentially, isn't it? Is that correct?
No, rather, we are sure at this point, we are fairly confident that the deletions we've put in are maintained, because we've already checked them. So this is to kill anything that doesn't have sigma F on, because sigma F is required for the expression of kanamycin resistance. So if we give kanamycin to them, only the ones with sigma F on can survive, and only the ones that are sporulating will have sigma F on. So basically we can select for sporulation.
So you're narrowing down the population so it's only the ones that you're interested in, essentially.
Yes, exactly. And this is we could check this under a microscope, and at this point we could take a little sample of the liquid culture, we could dye its membrane with a dye, and then we could put it under the microscope, and you could see the cells, you could see them. You've only got cells with these two small compartments, like we said before about the fact that there's two small cells in like a box in the middle.
Yes, yeah.
You can tell that the kanamycin has successfully wiped out any healthy cells that aren't sporulating, because you've only got cells that have got these extra compartments.
So this is where the mother cell has tried to essentially start this sporulation process twice, essentially.
Yes. Yeah.
Yes.
Yeah. And we've got this to start off with, the kanamycin. We tried a few different concentrations, because obviously we're designing an experiment, we don't know. And the first time that we did it, we thought, not all of the vegetative cells are dying quite. We should maybe up that a little bit. Well, let's use a bit more. And then when we looked at it under the microscope, they were unhappy. They were, their membranes were a little bit, you can tell if the cell is unhappy because it looks almost fuzzy under the microscope. It was really exciting, because I've been told if you do microscopy and you're not making mutants, it's apparently really boring, because everything looks the same and everything just looks like a healthy cell. But when I was making stuff, everything looked really unhappy and everything was really weird shaped.
There was lots, because I think I can picture the ones that because I think you showed me these photos, and there's lots of different variations of like lots of different weird shapes and patterns. Like obviously we can't actually describe them because this is a podcast, but it was there's lots of different details that you can analyze, and like the bacteria didn't enjoy that.
No, you're expecting almost a normal cells. You're just looking like a tic tac shape with, or like some long tic tacs with lines down them, and then you're getting the sporulating cells. You're getting like a rectangle with like a semicircle on the end, and then you've got the sigma E mutants, you've got 2 semicircles on the end, and then you've got that my sigma E mutants were curvy and really long and had little kidney bean shaped things growing off of them. And you've got so many permutations of what was going on. And it was fully just because we were letting them find their own way out of a harsh situation. So once we'd put this kanamycin, to go back to the process, they are growing healthily for 2 hours, then they are unhappy for 2.5 hours, and then after 2.5 hours of unhappiness, we add an extra thing to make them unhappy because we add the kanamycin. So, but that should only make the cells that aren't sporulating unhappy, because the kanamycin resistance is associated with sporulation. We've put it, it's got that tool if it is currently activating sigma F.
Yeah, it's associated with this sigma F factor that you need, that you're wanting to be on for your experiment,
Yes.
or for the cells that you're experimenting with.
Yes. So they had that kanamycin and salty water for half an hour, and then after this point, we transferred them to sugary water. Really nice environment, but with still kanamycin.
Okay. Yeah.
So what we're doing there is we are forcing. We've killed all the cells that aren't sporulating, and then we've put them in a positive environment. They can't go forward because they've not got sigma E, and they've got sigma F on. So this is where they are thinking, there's enough nutrients. I could start outgrowing, but my sigma F is on, so I've got to mutate something. The way we talk about bacteria and we say about mutating things, that's a little bit reductive, because mutations are not a reaction. They are spontaneous and then selected. You don't. A bacteria doesn't think, I'm going to change this. Bacteria have mutations and they go, oh, this is slightly different in me, and then, oh, I'm surviving better. Oh, everyone else has died. Oh, I'm going to continue to replicate. That's how it works, rather than.
Yeah.
It doesn't. It's not that. For example, natural selection. You see a giraffe doesn't see a leaf high up and go, I've got to get a longer neck. A giraffe that happens to have a longer neck goes, oh, I can reach this better. Everyone else is dying. Oh.
An advantage over the rest of the population, and so therefore it reproduces better and takes over the entire population. And
this is why the idea of replication and the idea of generational changes is why the culture that we used was continuous. So after we transferred it to the sugary water, we would keep it there for 2 hours, and then we would take a sample and put that on. We have liquid culture to grow fast, and then to look at things, we put it on solid culture. So petri dishes with like jelly in them. Yeah. So there's so much bacteria inside a liquid culture. So what you have to do is you have to dilute it on a logarithmic scale. So so taking 100 microliters of the culture, putting it in 900 of sugary water, and then taking 100 of that and putting it in another 900, and we were using dilutions of sort of -4 to the -6.
So that's 0.00001 to 0.000001. So very weak dilutions, essentially.
So we would take a sample and put it on these jelly, like the gel plates, and they could then, each individual bacteria overnight would replicate and grow a colony, and then you could see how many individual bacteria there were by how many colonies formed. So you'd have dots growing up on the plate.
Wait, so sorry, say that again. So you can tell how many bacteria there are from how much it's replicated?
So you can't see individual bacteria by the naked eye. But if you spread a sample of a bacterial population across a plate, every individual site where there is one bacteria will start asexually reproducing and form a kind of like a clump of cells. So if you dilute it far enough, it will be a countable number. If you did it from the start, it would just be, we call it a lawn because it's just completely covered. But at the factor of 4 or 5 diluting it, you can count 20 original cells or things like that. And that's really useful to we could use that to understand over time how many bacteria were surviving, because obviously we've put it through this difficult situation, and hopefully there should be an increase over time on how many there were, and we could plate the generation that we'd just put through the conditions against the starting population that we'd used and we'd put in the freezer. We ran 2 experiments side by side where the same conditions were being applied to both populations, apart from the addition of the antibiotic, which we didn't do to the control. So it was just to see how many cells there were, I guess, and then to compare them against each other. So it was to prove that there was a lot. It was to almost to prove that the kanamycin was working, because the control had a lot more cells in it. But also comparatively, you could see an increase in the cells treated with kanamycin by generation, but also it was still an entire factor of 10 smaller than if you didn't use kanamycin.
Yes, okay.
So you knew that you were killing any of the vegetative cells.
Yeah. Sorry, I just had a quick question. So
yeah,
you
absolutely.
mentioned that every day you're taking a kind of subset of the population to see how it's changed with time. What are you? Because surely if you're keeping the rest of the cells in the freezer, then they're not doing anything. You've almost frozen them in time in their growth. So what are you exactly measuring changing with time in this situation by plating it up on this petri dish and which is very dilute, and letting it grow to count, essentially? What are you exactly measuring with time?
So every day, we'd take a sample to look at the number. We would take multiple samples every day. So one sample that goes on a plate once diluted to see a rough idea of number, one sample that was mixed with glycerol to put in the freezer as a kind of backup, like a stock for if something failed. So we could go back and look at it. And it was almost like a snapshot. That's a freeze frame. So at any point, we could take that out of the freezer, grow it, look at it under the microscope. That's what it looked like that day. And then third sample, you could put on a plate, but this plate has kanamycin in it, which we will get to in a second. And then fourth sample, sorry, there are 4. Fourth sample is then transferred back into liquid media and grown overnight to form the next day's experiment.
Right, okay.
So that's why it's continuous. The idea that cells that survive from that day are then used and put under the same difficult conditions as the day before. It's got a repeated pressure.
Yes, okay.
So they're allowed to recover overnight. They are in sugary water. They are perfectly happy, but it's only the cells that survive the difficult situation that are allowed to recover. And then they're put in the difficult situation again. And hopefully each time they're put in a difficult situation, more of them survive because more of them are suited to the scenario. And then we can hopefully see an increase in the ability, because the only way that cells survive in this scenario, you've got that you're killing all the vegetative cells, you are forcing them to have sigma F on, and then you are putting them in sugar water. The only way that cells can grow overnight is if they outgrow from spores.
Isn't this how we were mentioning last time with this sigma A factor that originally in theory can't be on with sigma F, which is responsible for normal metabolism?
Well, this is slightly more complicated. There are I'd mentioned 4 samples.
Yes,
the counting them and the stock, they are for records, they are for tracking. But the 2 other samples, there are 2 different selection pressures there. So the one that we are taking away, we're taking a sample and growing it on kanamycin, that is a continuous pressure. There is still, if you take something that's been growing in kanamycin and you put it still in kanamycin, it has to have sigma F on, and to grow, it has to have sigma F and sigma A on, because of the fact that it has to mutate to allow the sigma F expression for kanamycin, but also has to have sigma A in order to replicate. So those ones that are growing on a plate, every generation we would count how many colonies could form on the kanamycin plate, so how many could have sigma F and sigma A. However, the ones that are used for the next day, they are allowed to recover without kanamycin. So they are removed from the antibiotic and put in sugary water with nothing else. So though we've killed everything that's not suitable, they're then just able to do whatever they like. So it's a lot. They can then probably, hypothetically, they're turning sigma F off, or they're going, because they're outgrowing, they don't need sigma F on anymore. They are able to reverse the turning on of sigma F and just go back to sigma A and normal metabolism.
Okay, yeah.
The next state of culture can only come from cells that outgrew, because we killed everything else with the addition of antibiotic. So every population is being formed from successful outgrowth.
Yeah, okay.
Because of and that means that they must be, every generation is being formed from uncommitted cells.
Yes, yeah. So because so both populations, not the records ones, but both populations you have, both of them are formed from outgrowing cells. It's just the first one that you mentioned is because you're putting it in the kanamycin, you're having to they have to keep sigma F on to stay alive. So that outgrowth is when we have when we have sigma A and sigma F on at the same time, and then the next day cells, that is outgrowth, but then that is not necessarily having sigma A and sigma F on at the same time, because it's not in kanamycin, it's just in sugary water, and therefore it can turn sigma F off and survive. Essentially, it's not having to keep
because
sigma
it's
F on.
Because it's able to uncommit, it should be able to get rid of the sigma F when it doesn't need it on anymore, because it doesn't need kanamycin resistance. So it's able to just reverse, it's able to turn around, but it's had to have that kind of, it's had to have sigma F on and start outgrowing with the presence of sigma F. Because it was in kanamycin earlier, it was just that it kind of is able to have a head start and then is transferred to a much more pleasant environment to replicate long term. Yeah, the two selection pressures, as you'd say, are the continuous culture. So the one that feeds into the next day is selecting for healthy outgrowth, because only the cells that form the first stages of sporulation and then start outgrowing can repopulate the population, because all of the other cells all healthy cells have been killed, all vegetative cells have been killed. And then you've put them in an environment where they should be able to grow, but they should also be stuck unless they can outgrow. So only outgrowing cells repopulate for the next day.
Yeah. Okay.
The second selection pressure, the idea of the idea of placing them on kanamycin, that's not continuous. That's at every generation we see the proportion or the output that is able to survive on kanamycin, and that is selecting for constitutive sigma F expression, the ability to continue to have sigma F expressed whilst growing.
Yes, okay.
Which is the idea. This is the kind of counterintuitive, these two things shouldn't really exist at the same time. And this was something that we weren't sure that this was going to happen. My PhD student and her supervisor had differing theories on how it would work. She thought that the continuous process would be the only successful one, and that if we continued to grow them on kanamycin, nothing would grow because they couldn't have both on. And my supervisor's supervisor thought that the continuous culture would not select for outgrowth effectively enough, and that we would need to force constant sigma F expression in order to properly cause the mutations. They were both right and both wrong, because both worked. But in different ways. We got different results from the different pressures, which is to be expected. But we weren't sure. To start off with, the kanamycin, we were taking every day, we were taking 7 samples, 7 200 microliter samples of this culture, and putting it on a kanamycin, 7 different kanamycin plates. And then every day I was looking at 7 kanamycin plates that were completely empty.
Okay.
And I was thinking, oh, it's not going to work. This is never going to work. This is really sad. And then day 3 was over the weekend. We left the cells for longer. We left the cells for 2 extra days, and they started coming up with these tiny little dots. And we were like, we were just thinking, surely not in 3 generations if they decided to mutate to the point where they can just have 2 sigma factors on that actually oppose each other's functions. this is amazing.
Just generally a cool result from the experiment that they can have. I guess that you found that they had these two things on at the moment. And so if I'm correct in saying you're almost testing for these two conditions that you were testing for. One was this permanent kanamycin, so that they had to have these two different metabolisms happening at the same time, which is what you obviously found after this day 3. And then the second one was testing for healthy outgrowth, but after the sigma F factor, after it had been placed in kanamycin, essentially.
Yes, so the ability to repopulate a culture. So the thing we're selecting for, you could see in the appearance of the mutants when we looked at them under the microscope, and when we had to try and grow them, you could tell the difference between the selection pressures immediately, because there was a completely different priorities in these cells. Almost the selection pressures, the way that the things that they were, again, I'd say the word choices like they were thinking, but the sacrifices that were made in their kind of survival were different.
Okay.
The kanamycin plates, every day we would, over the time, obviously day 3 was the first one, it was very slow to appear, and there weren't that many. There were like maybe 20 on a plate. By the end, we were getting hundreds on a plate overnight, so it was getting quicker, and it was getting, so this one wasn't continuous. It was just that every generation we would see if there was an increase. They weren't being used to try and repopulate. They were just being taken out and looked at.
Yeah, okay.
But they're the same cells. It was just out of the cells in a generation, how many can survive being in kanamycin permanently? And then out of each generation, how many can healthily repopulate a new generation, which are two completely different functions, because if something is being forced to have sigma F on constantly, it's being asked to sacrifice everything in order to, or anything it needs to keep that resistance cassette on.
No, but then it's the sigma A is the normal metabolism that you'd expect in vegetative cells.
Some of the sacrifices in order to maintain growth whilst being resistant to kanamycin were not ones that we'd want necessarily. You could see a lot of the mutants that we looked at completely lost the ability to form spores.
Oh, because they're having to keep these two functions on at the same time?
Yes, so in order to keep sigma F on constantly, they had almost removed its function in sporulation so that it could, sigma F was then just there purely to provide kanamycin resistance rather than to start the process of sporulation.
So you're saying that if you tried to then almost begin the sporulation process again by putting them in, say, really harsh conditions, they would just die. They wouldn't be able to sporulate.
Well, they would, some of them, be able to survive but not form spores and then die over time. They would. So what would happen? The number of ones. So what we'd do is if they grew on a kanamycin plate, we could then take a colony and then put it through the same conditions as the continuous culture. So you're growing it for 2 hours, you're putting it in the sporulation media for 2.5 hours, you're putting it with kanamycin, and then you're transferring it to sugar water.
Yeah.
That was basic. That is the controlled. Those are the growth conditions. And for everything that we looked at under the microscope, we used those growth conditions because that ensured that everything had gone through the same pressures.
Okay.
So we would use that to look at the generations that we've been using to form continuous culture, but also use that to analyze the ones that were turning up in these kanamycin plates. Some of these you'd put them in the sporulation media and they would start dying. You look at them at 2.5 hours, or you look at them at 3 hours, you look at them, you've added kanamycin. Some of them, they've survived enough to grow on the kanamycin plate, but when you put them back in kanamycin, they don't want to be put back in kanamycin and they die. Or some of them don't know how to form spores anymore because they're so reliant on sigma F as a provider of a resistance cassette that they've turned off. Again, this is an active area of research, and we are looking only at phenotypes and not genotypes, because at the end of my project, we were able to send off some of the samples for genetic sequencing, but that was the end of my project. So my PhD student will use this to hopefully further her research, but it wasn't part of my project itself. So all of the things that we talked about were hypothetical due to our knowledge of what genes do and how different processes are controlled, but we cannot be sure. But the concept that we, I guess, hypothesized was that maybe in order to have sigma F on in vegetative growth, which they needed to survive being put on that plate, they had removed something that activated sigma F as part of the sporulation process, so that they could just activate sigma F all the time.
So they were able to have this kanamycin resistance for the normal growth, but if, say, you put them in a medium with kanamycin and, say, salty water, so you also remove the nutrients, then they would die because they've lost that process to sporulate, which is obviously what they would normally do for like completely normal cells in this salty water, because it's harsh conditions, it's non-favorable conditions.
Mm-hmm. Well, we looked at them when we were looking at them. You'd look at them at the end of the sporulation condition, so the end of those 3 hours in salty water, and then you'd look at them an hour in sugar water and then 2 hours in sugar water. So what you'd see with the cells that had lost, what we assume lost the ability to sporulate because of some, I guess, prioritizing sigma F expression over everything else, you've got that they would look really, really unhappy. You'd get wiggly cells with holes in their membranes, but they couldn't form spores at all. And then you'd put them back in sugar water and they'd just start growing again.
But and that is that one of the reasons why you assumed that these cells have kind of sigma F expression for normal vegetative growth?
Yes.
Because even though they can't form spores, they can still perform this normal healthy growth in sugar water.
When they are exposed to kanamycin, they are resistant, so they must have sigma F switched on.
Yeah.
But they are not forming spores because you're not getting these little cells forming. You've just got normal cell division. So they've lost the ability to start that cascade.
Yeah. Okay.
But we know they're using sigma F because that's the only way they can be surviving the resistance.
The kanamycin, yeah.
Yeah, that's the only way they can be surviving the antibiotic.
Oh, okay.
But that's not the only thing that happened. The thing with this kind of, the idea of putting things in kanamycin, we were producing mutants that looked and acted a number of different ways. And this was really exciting because it meant that we knew we had influenced the cells and potentially multiple different genes related to commitment because they were acting in different ways.
Okay. So how are they acting? Go on.
So you had some that had lost the ability to form spores. You had some that formed these kind of 4 spores and then didn't grow. You had some that grew really long, some that were able to grow but seemed to grow in the wrong shape, some that formed way too many lines down them and were dividing too many times, and things like that. There was some strange problems that implied something had happened to the membranes, which was interesting because this is something that when looking at the sequencing, we will hopefully be able to understand what exactly was going on. But basically with this condition, we'd managed to create a load of mutants that we weren't selecting for healthy outgrowth, we were just selecting for keeping sigma F on. So they were changing anything they could to keep sigma F on. And that was leading to abnormal spores or abnormal vegetative growth and not really successful. It was interesting,
Okay. Yeah.
but it wasn't necessarily, these cells weren't healthy. They took longer to grow. They were really, really hard to transform because they weren't healthy enough to take up DNA to tag proteins to look at colors, so to use fluorescence to tag things. It was really hard to convince them to do that. Only a few of them did.
Okay.
But when they grew on the plates, even, they looked different. They were the colonies, the little blobs that kind of form, they were a different texture and sometimes a different color and a different size to the ones that you would expect.
Oh wow, okay. And am I right in saying that this is due to the genes that were under sigma F? These are the ones that you were kind of hoping would change, or hoping would change from the mutations, because you were forced, because as you mentioned, you were selecting to keep sigma F on.
Well, yes, we don't know what the mutations are. We can only describe phenotypes because we can't tell what the genes were up to, really. But
yeah, you need the
yes,
gene, not screening, what is it?
Sequencing.
Sequencing. Yeah, you need the gene
Yes.
sequencing to decipher the genotype.
Yes, but we know that whatever mutations were happening caused significant changes to morphology and phenotype for both sporulation and vegetative growth, because what I did once we were getting consistent growth on these kind of mycin plates, every day I would take 32 colonies and grow them all individually and then look at what they looked like on the plates. And then we picked a good number of those and looked at them under the microscope as well. And we were looking at what they looked like. So some of them they were almost waxy in texture when growing on the plate, when they would normally be matte. And some of them had slightly pink edges, or they were a lot more dome shaped than they should be. And we were taking for generations 4 to 10, there were 32 different samples where I was writing down how these different different organisms were kind of acting, and then we could look at some of those under a microscope if they grew strongly enough. That was the problem, that they were quite unhealthy. So when we would grow them to look at under the microscope, sometimes we couldn't get a large enough population to look at them. But when we could, it was fascinating because they were mutants. They were the exact stereotype of what you think of when you think of the word mutant. These things were not happy. They were not healthy. They were surviving because they could, but they were not thriving. None of them really were thriving. They were all abnormal in some way or other.
And so obviously there's, and as you described, there's obviously some very different mutations that are going on. Obviously you were only selecting for keeping sigma F on to survive the kanamycin. So is that basically telling you that there are many different mutations that can do that because there are so many different types of behavior? Because presumably if there was one very specific mutation that would allow this kanamycin to be specifically to be kept on at all costs, then you would only see the same type of behavior for all of the different cells. But there's obviously many different ways and different mutations that can do that, essentially, because of all the different weird abnormal behaviors that these cells were undergoing.
Yes, what we were thinking there was the loss of sporulation, the idea that some of them would simply revert to vegetative growth. That was likely to be the easiest way out, I guess. And that was maybe the kind of the almost separating sigma F from sporulation and just using it as a resistance, and then prioritizing the ability to resist and saying, I don't need to know how to sporulate anymore. I just need to be able to survive this kanamycin. That was the kind of the theory that we had decided, that we kind of used to describe this activity that we were seeing. But the other ones, the ones that were outgrowing, but strangely, the promising thing there is if we took some of these different phenotypes, we could sequence all of them, and hopefully they would be caused by different mutations of different commitment factors. And then we would be able to find those and combine them in an artificially created mutant that would then be much better at outgrowing than the ones that we'd previously created.
So here you're talking about those out of those 100 genes that were under sigma F, these specific ones that are related to commitment, and by analyzing these, not only the gene sequencing, but also analyzing these different mutants, you can identify essentially what specific genes are related to each aspect of this commitment process, essentially.
Yes, we don't know, obviously, as I haven't looked at the sequencing, I don't know. There's a potential that the mutations that occurred weren't ones that we wanted, but the way that we've created the environment, the idea that they have to keep sigma F on, but they can't use sigma E, if they were able to start that sporulation process and they were able to outgrow, the chances are any mutations that occurred were to do with commitment because their ability to uncommit improved.
Yes. Yeah.
But no, these cells were wildly unhealthy, especially in comparison to the other selection pressure. The idea of continuous culture, these repeated generations where we took the healthiest cells to populate the next generation, we were selecting for, we weren't too fussed about how good they were. Obviously, they were using sigma F to form spores and they were using sigma F to resist kanamycin, but they were only asked to use sigma F for kanamycin resistance as a sporulating cell. And then once they were no longer sporulating, they were removed from that kanamycin and they were allowed to start vegetative growth again from successful outgrowth and populate a new generation. These we could compare over the generations, and by the end of the experiment, we had created consistent increased outgrowth. So there were, at the end, we took the last healthy generation was generation 9 because generation 10 didn't seem to be too happy. We looked at the end, we looked at generation 7, 8, 9, and 10. And by the end, we decided, we looked at them all under the microscope, and the most promising actually was generation 8. Sorry, the generation 8 had the largest outgrowths, and it was very healthy. And you could tell that we were hoping that these were outgrowths. Obviously, at this point, we didn't absolutely know, but you could see there were the smaller cells were growing in size, and there were a number of cells that looked like vegetative cells at later time points. But we knew that we'd killed all the vegetative cells, so they must have been healthily growing cells that had just outgrown and continued to live.
I suppose that makes sense as well, because obviously the ones that you were constantly keeping, the cells that you were constantly keeping in kanamycin, obviously they are more likely to develop mutations to keep sigma F on at all costs because they are constantly in kanamycin. But the ones that you are putting in kanamycin, but then you're after that putting in ideal conditions, which is the sugary water, I guess it's more likely, looking at it from that way, that they're likely to develop mutations that aren't abnormal because they're not put in as much stressful conditions or stressful conditions all the time, if that makes sense.
Well, yeah, the selection itself was for, rather than the selection being for sigma F, the selection was for health, because only the ones that survived and only the ones that grew well would be used in the next day's culture. So you were only, the next day's population was built of the healthiest cells from the previous day. So you were promoting successful and healthy outgrowth, which was a much more, we could use this to, we could see that these cells were similar to those of the triple mutant or of sigma F deletion. They were somewhere in between. All of the generations we looked at for this selection pressure were somewhere between the original starting point and a control that we had of a sigma F deleted mutant that was able to fully outgrow. So they were exhibiting a level of uncommitment between the starting point and the ideal point.
Yeah, so it's
depending on the generation.
It's this idea of partial commitment, but it's just having, because I think I now better understand why you had this control, or not the control, sorry, but the other condition where you're kind of switching it between like kanamycin and salty water and then sugary water. Is because you're trying to exhibit in these cells some level of partial commitment, but you're having two different priorities for the mutation, essentially. One being the sigma F factor being kept on at all costs, and the other being we need to prioritize healthy growth because that's what you're taking as the sample for the next day is the healthy cells.
Yes. And
Okay.
not only was that useful for creating the next day's generation, but it was also we were able to take at the end point of the most successful generation, generation 8, we could look at that and take samples of this and start to look at what that outgrowth looked like and what those cells looked like at the different stages under the same conditions under the microscope as the kanamycin resistance selection mutants, and to compare how they were looking under this selection pressure to those that under the other selection pressure. And again, you were seeing a different phenotype, so hopefully a different mutation. And then the idea would be that you can find all of these mutations and you can test them for their relevance to commitment, and you can, if successful, combine them to create an
artificial mutant
that's even closer to full uncommitment, or maybe even at full uncommitment, depending on how much we've found out.
Yeah, so it's genuinely really fascinating. I think something that also kind of was, I guess, indicated to me in this episode particularly is that when we typically think of mutation and evolution, we think of it happening with, I guess, mammals on extremely long timescales, like millions of years. But I guess this is the advantage of working with bacteria, because bacteria are so fast at mutating and adapting to their environment. And this is something that is clearly, I guess, kind of indicated with this study, because you can get them to mutate and adapt to conditions on like a timescale of days, pretty much.
Yeah, the only reason we were able to get them to mutate was because we put them in such a horrible place to start off with.
They literally had no choice.
Obviously, it was really useful that it was such an easy thing to work with. But yes, we designed an environment so tricky for them that they had to mutate in 3 days. Yes, is another way of wording it, I guess.
Yeah.
So we were torturing them, but
we'd like to pass
for the good of science.
We'd like to pass on our thoughts, not only to the amount of bacteria that you've murdered and tortured throughout your experiment, but also, I guess, the people who are going to be doing future experiments who are not going to be working with such an easy bacteria like Bacillus. You made one final thing I had. I don't know how much you can talk about it, because I appreciate that it's an active area of research, and also your PhD student, she hasn't released her thesis yet, but you mentioned about this thing with like creating an artificial mutant. Is that actually something that she's going to do in her PhD, or is this like is this like more research in the future that you're talking about?
Well, this is the idea. My starting point for the experiment was her previous artificial mutant, this triple mutant with sigma F on and sigma E off. The triple mutant, the most uncommitted model, is a sigma E deletion with 3. Well, by the time that I was working with her, she was starting to work on a potential 4th. But what was ready for when the start of my experiment was a triple mutant with 3 deletions that exhibited partial uncommitment when these 3 deletions were put together. And it's the idea that any genes that come out of my experiments as potential commitment factors could be added to that triple mutant to create a quadruple mutant, or like even up to 7. We don't know how many it is, but to create a further
even less commitment
mutant. So the aim in general is to create a mutant that is fully uncommitted. It looks the same as sigma F does, but it's got sigma F on.
Yes.
Sorry, same as a sigma F deletion mutant, but it's got sigma F on. So hopefully we will talk in the next episode about the outcomes of this experiment. Now we've talked about the selection pressures. Where did that lead me? I've talked a little bit about it, but I'm going to be able to hopefully reflect a little bit next time and talk to you about where the sorts of things that we managed to see and what that might mean for the future. Again, secretly, without any gene names, because
we can't say. But yes, we shall leave that till next time. That's a bit of foreshadowing for what we talk about in our last episode of completely and
Yes.
utterly sporing. But yes, it was good. It was, and certainly we've talked about some very significant results that you've obtained that, as we'll talk about in the next episode, can go on to more research in the future, which is pretty cool.
Yes. Hopefully. Yeah. It's quite exciting to feel
if
like
you're
I've
a
actually
nerd,
achieved something.
but like
Yeah, if you're a nerd.
that is pretty cool. But it is very cool though, and you're right, it is a huge achievement.
I mean, hopefully it leads somewhere, but we will see. And the next episode of Completely and Utterly Sporing.
Spoiler alert, it probably will, but yes. Join us in the next episode though, because there's more to that. Okay.
Thank you.
Bye.